Good evening, everyone. Today, I want to bring your attention to something that often escapes conscious thought, but deeply shapes how we feel, what we do, and even who we're drawn to. I'm talking about smell, taste, and pheromones, three sensory systems that don't just reflect the world around us, they drive our internal states and behaviors. Let's start with smell, or more precisely, the olfactory system. What's fascinating about smell is that it's the only sense that bypasses the usual relay station in the brain, the thalamus, and goes straight to the amygdala and hippocampus, the centers of emotion and memory. This direct connection explains why a single whiff of a scent can instantly trigger vivid memories or deep emotional states. A smell isn't just a smell, it's a neurochemical access point to who we were, and sometimes who we become. Now, let's move to taste, which is far more than just pleasure. It's a survival mechanism. Sweetness tells us about energy, rich glucose. Saltiness helps us regulate hydration. Sour and bitter, they're defensive signals, often indicators of spoiled food or toxins. What's remarkable is that these taste signals don't just inform us, they bias our behavior. Under stress, for example, we often crave sweet and salty foods. That's not a failure of willpower. That's a deep evolutionary circuit designed to keep us alive during threat. Then there are pheromones, chemical signals released by the body that subconsciously influence the behavior of others. Now, in humans, this is subtle, but the data is clear. Pheromones can impact mood, sexual attraction, hormonal cycles, and even interpersonal trust. This happens through the vomeronasal organ and other olfactory pathways that influence parts of the hypothalamus involved in reproduction and bonding. You're not always aware of it, but your behavior, especially in social and intimate contexts, is being shaped by these invisible chemical cues. All of this works through what we call neuromodulation. Smell and taste don't just give you information, they actually change the levels of key neurochemicals in your brain: dopamine, serotonin, oxytocin. That means a smell can make you more alert, more relaxed, more social, or the opposite. The impact is real, time, it's measurable, it's behavioral, and it doesn't stop there. Sleep quality, learning, immune function, all are tied to olfactory and gustatory input. Studies show that certain scents, like lavender, can enhance deep sleep and even memory consolidation. Taste, through its interaction with the gut-brain axis, can signal immune readiness. What you smell and taste literally changes how your brain and body prepare for the next challenge. Finally, perhaps most empowering of all, these systems are plastic. That means they can be trained. You can become more sensitive to beneficial smells, more discerning of taste, and more attuned to your own behavioral shifts in response to chemical cues. With awareness and repetition, these systems can be used not just to survive, but to optimize how you live, learn, and connect with others. So the next time you encounter a scent or savor a flavor or find yourself inexplicably drawn to someone, recognize that you're experiencing ancient, powerful systems at work. They're not beneath consciousness, they precede it. And by understanding them, you begin to take control of something incredibly fundamental: how you feel, how you act, and how you connect. Thank you. The olfactory system is unique among our senses because of the directness of its neural wiring. Unlike sight, sound, or touch, which all pass through a central relay station in the brain known as the thalamus, smell takes a shortcut. It bypasses the thalamus entirely and connects directly to the amygdala and hippocampus, two of the brain's most ancient and evolutionarily conserved structures. These regions are critically involved in processing emotion and memory. The amygdala assigns emotional significance to incoming stimuli, especially those related to threat, reward, or safety. The hippocampus, on the other hand, encodes and retrieves memories, particularly those tied to context and experience. Because of this direct pathway, smells can provoke powerful emotional responses and vivid, often long, forgotten memories almost instantly. A faint scent of a perfume can bring back the memory of a person you haven't seen in years. The smell of fresh, cut grass might trigger emotions tied to childhood summers. These reactions are not just poetic, they are neurological. The brain binds smells to emotionally charged experiences much more efficiently than it does with most other sensory input. This is why scent is often used in therapeutic contexts, from treating trauma to enhancing learning, and why it plays such a profound role in shaping our mood, attention, and even decision-making. In essence, when we perceive a smell, we are not just identifying a chemical in the air, we're accessing a direct portal into the emotional and memory architecture of the brain. This makes olfaction not just a sensory tool, but a behavioral driver capable of altering state, biasing perception, and influencing action within milliseconds. Taste, while often associated with pleasure and indulgence, evolved first and foremost as a survival mechanism. It is the body's built-in chemical evaluation system, allowing us to rapidly assess whether something is nutritious, safe, or potentially harmful. Each of the primary taste categories, sweet, salty, sour, bitter, and umami, corresponds to specific types of compounds that signal value or risk. Sweetness typically indicates the presence of glucose, the brain and body's preferred energy source. Saltiness reflects essential electrolytes needed for fluid balance and nerve function. Umami, the savory taste, suggests the presence of amino acids, which are the building blocks of protein and necessary for tissue repair and neurotransmitter synthesis. On the flip side, sour and bitter tastes often serve as warning signals. Sourness can indicate spoiled or fermenting food, while bitterness is commonly associated with plant toxins or alkaloids that can be harmful in large quantities. These taste responses are deeply rooted in evolutionary biology, honed over millions of years to help organisms distinguish between what will sustain them and what might harm or kill them. Beyond the biochemical signaling, taste also influences behavior through powerful neural pathways. The act of tasting activates regions of the brain involved in motivation and reward, particularly the dopaminergic system. This can drive food, seeking behavior, reinforce certain eating habits, and even shift emotional state. In times of stress, for instance, the body often craves sugar or high-fat foods, not because they're simply enjoyable, but because they provide fast energy and activate neural circuits that reduce perceived threat. Importantly, taste is not static. It can be conditioned and modulated over time. Preferences can shift based on nutrient needs, hormonal state, learning history, and even gut microbiota composition. This makes taste a dynamic interface between the external world and internal physiology. So while we often think of taste as a form of enjoyment, at its core, it's a tightly regulated biological system designed to keep us alive, balanced, and behaviorally tuned to what the body needs at any given moment. Pheromones are chemical signals released by the body that play a subtle but powerful role in shaping human behavior, particularly in social and sexual contexts. Although we often associate pheromones with animals, insects finding mates, dogs marking territory, the human body also produces these invisible signals, and our brains are equipped to detect and respond to them, often without conscious awareness. Unlike smells we can easily identify, pheromones tend to operate below the threshold of conscious perception. They don't trigger recognizable odors. Instead, they act directly on the limbic system and hypothalamus, regions of the brain responsible for regulating instinctive behavior, emotional responses, and hormone release. Research has shown that pheromones can influence a wide range of interpersonal dynamics. For example, they can affect sexual attraction, with individuals often feeling drawn to partners whose pheromonal profiles are biologically complementary, particularly in terms of immune system compatibility, as detected through the major histocompatibility complex MHC. In group settings, pheromones may subtly shape feelings of trust, dominance, or synchronization. One of the more well-known phenomena, though still debated, is menstrual synchrony, where women living in close proximity may experience shifts in their menstrual cycles that bring them into alignment, likely due to the influence of pheromonal cues. These effects are not magical. They are mediated by specific receptors, such as those in the vomeronasal organ and in the olfactory epithelium, which send signals to parts of the brain that regulate endocrine function. This means that pheromones can change levels of testosterone, estrogen, cortisol, and oxytocin, all hormones that influence mood, arousal, bonding, and stress reactivity. Importantly, these changes don't require visual or verbal cues. You might feel more relaxed or more alert around a certain person, more trusting or more guarded, and often the explanation lies in a set of chemical interactions occurring far below the level of your conscious reasoning. In essence, pheromones are part of a silent communication system, one that evolved long before language and continues to operate alongside it. They don't override our choices, but they do nudge them, often strongly, in one direction or another. Understanding their influence gives us a deeper appreciation of how our environments, and the people in them, shape our behavior in ways that feel intuitive but are deeply biological. Neuromodulators like dopamine, serotonin, oxytocin, and norepinephrine are chemical messengers that regulate brain states and behavioral outputs, and they are highly responsive to sensory input, particularly scent and flavor. Unlike neurotransmitters that work locally and rapidly between neurons, neuromodulators influence broad neural networks, adjusting how the brain processes information, assigns value, and generates motivation. What's striking is how quickly and precisely these modulators can be shifted by something as simple as a smell or a taste. For example, the scent of food can trigger a surge in dopamine even before a single bite is taken. This is part of the anticipatory reward system, the brain's way of preparing the body for action. Similarly, the actual taste of sweet or fatty foods leads to further dopamine release in areas like the nucleus accumbens, reinforcing the behavior and increasing the likelihood of repeating it. This mechanism helps explain why certain flavors are so habit-forming and why food can quickly become linked to emotional states like comfort or relief. Oxytocin, often called the bonding hormone, can also be released in response to scent. Smells associated with loved ones, familiar places, or nurturing environments can trigger oxytocin pathways, increasing feelings of trust, emotional warmth, and social openness. These responses are not just psychological. They are biochemical changes that alter how we perceive others and how we interact. In romantic or intimate settings, for instance, the combination of pheromonal signals and familiar scents can enhance oxytocin release, deepening feelings of connection and attachment. Serotonin and norepinephrine, two neuromodulators tied to mood, arousal, and focus, are likewise affected by taste and smell. Certain smells, like citrus or mint, can increase alertness and improve cognitive performance, while others, like vanilla or lavender, promote relaxation and stress reduction by modulating serotonin levels. On the gustatory side, foods rich in tryptophan or omega-3s can support serotonin synthesis, further linking flavor to emotional balance. All of this happens in real time. When we encounter a smell or flavor, neural circuits adjust almost immediately to guide our attention, shape our emotions, and bias our decisions. This makes scent and flavor not just passive experiences. but active levers we can use to shift brain chemistry and behavior on demand. Whether it's choosing the right smells to support focus and sleep, or leveraging taste to reinforce healthy habits, these sensory cues are powerful tools if we know how to use them. Smell and taste play a far more integrative role in human physiology than we typically recognize, extending their influence well beyond the immediate experiences of flavor and aroma. In fact, both senses have been shown to directly affect key domains such as sleep quality, cognitive function, and immune regulation, largely through their impact on brain state and systemic signaling pathways. This connection underscores how sensory input isn't just informative, it's regulatory. Take sleep, for example. Specific scents such as lavender, sandalwood, and chamomile have been demonstrated in clinical studies to activate the parasympathetic nervous system, the branch responsible for rest and recovery. These scents reduce heart rate, lower blood pressure, and decrease cortisol levels, collectively priming the body for deeper, more restorative sleep. The olfactory system's direct connection to the limbic brain structures like the amygdala and hypothalamus helps explain why these effects are so pronounced. When used consistently, certain smells can become conditioned signals for sleep onset, effectively training the brain to shift into a more relaxed state faster and more reliably. The relationship between smell, taste, and learning is equally compelling. Sensory cues can influence how effectively we encode and retrieve information. Research has shown that exposure to a particular scent while learning a task, and then again during sleep, can improve memory consolidation, a process heavily reliant on hippocampal function. This is because scent cues can help tag memories with stronger contextual associations, making them easier to recall later. Taste, through its interaction with gut, brain signaling, and dopamine release, also affects cognitive performance, particularly motivation and attention. The nutrients and compounds associated with certain tastes can enhance synaptic plasticity, the underlying mechanism of learning and adaptation. As for the immune system, both smell and taste have powerful downstream effects. The gut, which houses a significant portion of the body's immune cells, responds to flavors and food compounds in ways that affect inflammation, microbial diversity, and immune readiness. Bitter compounds, for instance, have been shown to activate certain immune pathways, preparing the body for potential threats. Likewise, the mere anticipation of food, triggered by smell, can initiate salivation, enzyme secretion, and immune priming in the digestive tract. Furthermore, olfactory receptors have been found not just in the nose, but throughout the body, including in immune cells, where they appear to play roles in detecting pathogens and modulating response. What all this illustrates is that smell and taste are far more than passive reflections of our environment. They are active participants in shaping internal state. Whether it's influencing how deeply we sleep, how effectively we learn, or how robustly we defend against illness, these senses help regulate some of the most fundamental systems in the body. And crucially, because they are accessible and trainable, they offer practical leverage points for improving overall well-being. Our chemical senses, smell and taste, are not fixed or static. They are highly adaptable, shaped continuously by experience, environment, emotional state, and even intention. This plasticity means that the way we perceive and respond to smells and tastes is not just a biological given, but something that can be trained, refined, and redirected over time. From a neuroscience perspective, this is a function of how the brain updates sensory maps and neural associations based on exposure, repetition, and relevance. For instance, many people find the taste of bitter vegetables like kale or Brussels sprouts unpleasant at first, yet with repeated exposure, especially when paired with positive contexts, like a rewarding meal or health goals, those same flavors can become not only tolerable, but even enjoyable. This shift happens because the gustatory system is tied to both reward circuitry and memory systems. When the brain starts to associate a particular taste with a beneficial outcome, whether it's feeling better, achieving a fitness goal, or social bonding, the response to that taste changes at the neural level. This is an example of sensory plasticity in action. The same principle applies to smell. A scent that once triggered anxieties, the sterile smell of a hospital, can, through deliberate retraining or positive reassociation, become neutral or even calming. In therapy settings, scent is sometimes used in exposure techniques to help individuals rewire emotional responses to past trauma. Athletes and performers have also been known to use specific scents as performance anchors, smells that, through repetition, become linked to focus, energy, or calm. Context plays a massive role in this. A smell or taste experienced during stress or fear may be perceived as aversive, while the same stimulus, when encountered in a relaxed or joyful state, may be interpreted as pleasant or nostalgic. This context dependence reflects the brain's constant integration of internal and external cues to guide behavior. The environment in which a sensory input is received, who we're with, what we're doing, what we expect, all modulates how the brain encodes and interprets it. Because these systems are trainable, they also offer a unique opportunity for behavioral design. You can condition yourself to associate certain flavors with focus or reward, or train your nervous system to respond to specific smells with relaxation or alertness. This is not hypothetical. This is basic associative learning backed by robust neural plasticity. With intention and repetition, the chemical senses can be shaped to support your goals, regulate your mood, and optimize your performance. In essence, smell and taste are not just inherited sensitivities. They are dynamic, malleable systems that reflect how we interact with the world and, more importantly, how we choose to shape those interactions over time. In conclusion, the chemical senses of smell, taste, and pheromones are far more than simple physiological functions. They are deeply intertwined with our behavior, emotions, and overall well-being. Smell, with its direct connection to the brain's emotional and memory centers, can trigger powerful emotional responses and vivid recollections. Taste, originally designed to guide survival, continues to shape our eating behaviors and decision-making, influencing everything from our cravings to our cognitive performance. Pheromones, though subtle, influence social dynamics and sexual attraction, often guiding behaviors in ways we're unaware of. Neuromodulators, such as dopamine, serotonin, and oxytocin, respond to scent and flavor in real time, adjusting our brain chemistry to better suit our current needs, whether it's relaxation, motivation, or social connection. Smell and taste also impact our sleep, learning, and immune function, revealing how these senses affect not just our immediate experiences, but our long-term health and cognitive abilities. Most importantly, these systems are not fixed. They are trainable and context-dependent. With intention and repetition, we can shape how our chemical senses influence our behavior, turning them into tools for personal growth and optimal living. In understanding the power and plasticity of smell, taste, and pheromones, we gain deeper insight into how our bodies and brains work in harmony to guide us through the world. By becoming aware of these processes, we can harness them to enhance our lives, improve our health, and foster deeper connections with those around us. Ultimately, the chemical senses are a gateway not just to survival, but to a richer, more intentional experience of life. Thank you.